So today we're going to talk about RNA processing, and last time we talked about transcription, and I mentioned that there are many different kinds of RNA in the cell that serve different functions, and it turns out that in eukaryotes, and to a lesser degree in prokaryotes, RNA needs to be processed before it can go on to fill its functions. So today we're going to talk about different modes of processing in RNA and what that actually means for different types of RNA. Okay, so when people were first discovering DNA function and examining the relationship between DNA, RNA, and protein and uncovering the central dogma, it was at first thought that there was a 1-to-1 correlation between, well, I guess technically a 3-to-1 correlation between the nucleotide sequence in DNA and the sequence of amino acids. And so people were figuring out the structure of DNA, and that RNA is transcribed from DNA and is the intermediary for proteins, and that the genetic code of the DNA encodes amino acids, right? And so we soon realized that there is not necessarily a 1-to-1 correlation between the DNA sequence and the polypeptide chain sequence, such that lengths of DNA that are, say, 300 bases long don't necessarily yield polypeptide chains that are 100 amino acids long. And the idea of this direct correlation is colinearity between genes and proteins. And the reason that we don't have colinearity between genes and proteins in eukaryotes is that there are exons and introns within gene structure that mean that there are potentially large portions of the gene that are transcribed but are not translated. So so what you're seeing here are two genes that are, one is actually a mitochondrial gene, and the cytochrome B, and one is a nuclear gene, ovalbumin. And ovalbumin, what you're seeing here in light blue, are the introns. And you can see that in both of these genes, the introns are much larger than the exons. So the exons are numbered, and they're indicated in dark blue. So through the process of transcription and RNA processing, we end up with just the exons in the mature mRNA. Let's see if, okay, so as a clearer question, how many introns are present on a gene that contains 4 exons? And if you need to pause this and kind of go take a look at the slides, go back and look at that gene structure in the previous slide, you can go ahead and do that. So it turns out that the number of introns is directly related to the number of exons, with, as we go back here, the exons are interspersed with introns, and this is actually a very misleading figure. Interesting. So it turns out that there are actually 3 introns present on a gene that contains 4 exons, because introns are located in the interior of the exons. But I guess good thought exercise anyway, even though this figure is rather misleading in that regard. Okay, moving on. I'd like to say here that in bacteria, again, bacteria do not have introns, so they don't have RNA processing in the same way that that eukaryotes do. However, they do So transcription happens in the cell. Again, prokaryotes, remember, don't have nuclei. And so the circular chromosome is just in there in the cell. It's getting transcribed into RNA, and that RNA can then be directly translated. And in fact, I'll say this again when we cover translation, but in prokaryotes, transcription and translation can happen at the same time. Whereas in eukaryotes, transcription happens in the nucleus, translation happens in the cytoplasm, and so those two processes are spatially separated and therefore necessarily temporally separated. And so there is a lag between the time when a gene can be transcribed and the time when that RNA transcript can be translated. So in prokaryotes, we have an mRNA structure that includes regions that are not translated. So this is the mRNA that comes off of the gene. We have a region that doesn't get translated at all at the 5' end and at the 3' end, and these are called 5' and 3' untranslated regions, or UTRs. We then get a sequence in the middle that is translated. This is our protein coding region, and it is demarcated by the start codon at the 5' end and the stop codon at the 3' end. In prokaryotes only, there is a short sequence, a consensus sequence in front of the start codon that is called the Shine-Dalgarno sequence. That is all I'm going to say about it here. It is important for translation, which we'll talk about next time. But again, no introns in prokaryotic RNA, and this RNA can be translated directly. So the other interesting thing about that I failed to mention, I believe last time during the transcription lecture, was that in prokaryotes, prokaryotic genes are often organized one after the other, so they're clustered together, and we'll learn more about this as we cover DNA, or rather gene regulation in prokaryotes. And so often there are clusters of genes that are transcribed all together within a single transcriptional unit, so that a single mRNA transcript can encode multiple proteins from different genes. This is called polycistronic RNA, and I believe I mentioned it in lecture 12 notes, but I didn't mention it in lecture. So this is polycistronic RNA, and so this RNA can be cleaved to yield different RNA transcripts. I think also it can just be translated directly, so you have multiple ribosomes on a single mRNA translating different proteins. All right, so in eukaryotes, there is quite a lot of shenanigans that have to happen before what we call the pre-mRNA becomes the mature mRNA. One of these things is the addition of the 5' cap. And what you're seeing here is a schematic of what the 5' cap looks like. It is a chemical structure that's added onto the 5' end of the pre-mRNA. Here is what the chemical structure looks like. Don't worry too much about this. All you need to know, just general broad brush details, is that the 5' cap is a modified methylguanine. Whoops. So here's the guanine. Has a methyl attached to it, guanine with a methyl, and this is called a 7-methylguanine. It's bonded to a nucleotide here that's also methylated up here, and another nucleotide that's methylated here. Here the N is indicating that these can be any base, but there's this unique bond that's formed between the last nucleotide and the 7-methylguanine that is unusual for nucleic acid. So that's the 5' cap that's added on, and then we also get a poly A tail. So up here what you're seeing is the DNA. Then we get transcription. We get our pre-mRNA that includes a consensus sequence that is about 11 to 30 nucleotides upstream of a cleavage site. And this cleavage site is also upstream of a U-rich sequence. These signals indicate where the cleavage happens. So the pre-mRNA gets cleaved, and then you get the addition of a poly-A tail on the end of this. And this consensus sequence also serves as a polyadenylation signal for the enzyme that does the job. So we get the addition of 5' cap and a poly-A tail on the 3' end. And then we also get our intron spliced out. And intron splicing is kind of an amazing feat that eukaryotes have come across. And there are a number of signals that are required for proper intron splicing. There are consensus sequences at both the 5' end of the intron and the 3' end of the intron. There's also somewhere in the middle, but more towards the 3' end, is an adenine branch point. And what happens is that the spliceosome, which is this huge, it's one of the largest protein complexes in the cell. It's amazing. I think it has like over 300 components to it. It's ridiculous. It comes along and it first splices the 5' end, and then loops it around and connects it to the branch point. Meanwhile, it's got a hold of this exon 1 here, and it brings it closer to this exon 2 here, and then simultaneously splices the 3' end and then anneals the two exons together. It's an amazing feat. I posted a video kind of illustrating this in a more animated form, which I think will be easy to wrap your heads around. And so part of the process, part of the process of this intron, the 3' end joining, connecting with the branch point, it forms what we call a lariat, because it looks like a lariat, like a lasso type of situation. And the spliceosome has over 300 components. We're not going to worry about the vast majority of those. Just know that the spliceosome is another ribonucleoprotein. It's huge. It's got both protein components and RNA components, and some of the more important RNAs that are actually directly involved in intron splicing include 3 that are called U2, which is here, U2, U6, and U5. And the take-home message I want you to take home from this is that the RNA components of the spliceosome are critical for having complementary sequences that interact with the consensus sequences within the intron. So this is why in some parts, why consensus sequences are important, and why RNA can play such a big role in ribonucleoprotein function, because it allows the ribonucleoprotein to interact in a predictable and repeatable way with other nucleic acids, including DNA, as we saw with the telomerase, right? The telomerase has an RNA component that serves as a template in a predictable way because the sequence of the telomere is predictable. And so you can just make a single RNA component that goes into the telomerase, and it can extend any chromosome's telomere, right? So structure fits function yet again. And here in this case, we have RNAs that are annealing with the single-stranded mRNA. So we've got annealing with the branch point, annealing with the 3' consensus sequence in the intron, and annealing with the 5' consensus sequence, as well as with and crossing those intron exon boundaries to allow the two exons to come together, and so that when the 5' end here is spliced, then this releases the lariat. Well, actually, I think the spliceosome holds onto the lariat for a little bit, and then it releases it. But then it anneals the two exons. So it's a pretty amazing process.